if there's people who are listening to this who work in bioprocess, reconsider how you're using process analytical technology, especially with the advent of artificial intelligence. There could be huge benefits that you could gain from just starting to get a really solid, well-curated data set about your bioprocess. And a great way to start with that is use process analytical technology.
Hey, smart biotech scientists, welcome back. We're in the middle of a conversation about the art of communicating science, the potential impact of AI on the cell and gene therapy space, and our predictions for the biggest changes coming to the field over the next decade. Our today's guest is Benjamin McLeod, perhaps a familiar face to many of you from his insightful coverage of cell and gene therapy on LinkedIn. Ben currently works with NanoMosaic, an innovative analytical device company. How did you like part one of our conversation? Please hit pause now and tell us by leaving a review on Apple Podcast or the platform you're listening on. Your review means the world to me. Now coming up, part two of our conversation with Benjamin McLeod. Are you juggling the complexities of CMC development while trying to enjoy the beauty of biotech? Have you ever wondered if there's a way to simplify bioprocessing? Welcome to the Smart Biotech Scientist podcast, where we're diving headfirst into the very challenges you face. We're breaking it down, demystifying the jargon, and giving you the keys to unlock your full potential. I'm your host, David Brohlman, and I get it. With 15 plus years in the biotech industry, I face the same challenges you do. There's a way to simplify and streamline so you can remove complexity, you can skip trials and errors, deliver without delay your groundbreaking therapy to clinics and market, and still enjoy every single step. Do you want to learn how industry experts and I did it? Grab a cup of coffee and your favorite notebook and pen. Now is the time to take your bioprocessing game to the next level. Let's smarten up biotech. Now let's shift gear and let's talk about how AI will impact the cell and gene therapy space. So how do you see artificial intelligence impacting this space in the near future?
I had a really interesting conversation with someone yesterday about this. Just a brief conversation, even with regards to quantum AI that's coming, especially in the context of the biotech market seeming to pick back up again and there being a lot more investment. I am very intrigued by the ability of something like AI combined with quantum computing to truly map the millions, if not billions, of interactions that take place inside of a cell. And so by being able to do that, you've now got unprecedented access to manipulating certain pathways or seeing how different bioprocess conditions are going to impact cell viability, cell health, cell behavior, specifically in the context of bioproduction, right? Where you've got an established cell line, something like HEC293 or CHO cells or whatnot. You've got an ability now to actually map all of the millions of interactions taking place inside of that, and then use AI to find out what are the non-intuitive insights. What's the combination of 8 different factors that all applied at the exact same time or in stepwise fashion craft that optimal outcome, be it increased yield, be it better cell viability, be it whatever metric you're looking at. And so that ability to really dive down into the deep details and not just change one factor at a time, but actually assess the 3D space of biology in a far more precise and holistic fashion.
How do you think the combination of AI and quantum computing will impact the development of personalized cell therapies and then also the manufacturing? Because that's still quite a challenge to produce them in a cheap way and in an efficient way.
There's a really interesting company right now, I don't know if you've come across them, called In Silico Medicines, where they are using AI to identify small molecule candidates for various diseases. And they've already got progressing quite nicely through the clinic. Small molecules are kind of the early set of medications, but it will be coming for gene therapies and cell therapies that are a lot more complex. But that's the huge advantage here, right? Given that each person is a little bit different, and with a complex therapy like a gene therapy or a cell therapy, each person is going to react a little bit differently, even more so than with a small molecule drug. Just because, again, you're adding a complex medicine into a complex body. It's a little bit more simpler when you're adding a simple medicine into a complex body, or when you've got that added layer of complexity. If there is a way to use AI to map a personalized set of interactions and profile for a specific patient, and then correlate those specific patient profiles with these discrete or minute changes in the gene therapy or cell therapy that's getting made, it's going to be huge. But again, I'd say it's still a long way away, and it's going to be a lot more expensive before it gets cheaper, unfortunately.
Yeah, definitely. I mean, more technology or more sophisticated technology has a price tag, unfortunately. But are there any immediate benefits we could take advantage of and drive down the cost and make sure that a wider population of patients get access to these life-saving therapies?
Yeah. One aspect I'm very intrigued by is the combination of AI with process analytical technology, which I would say honestly still is slept on quite a bit, where first of all, people might only be looking at a handful of different parameters inside of their bioprocess and just kind of quickly correlate, okay, optimize pH to this level, and that's where you get a bump in yield or something like that. But if you are regularly testing 15, 20 different process parameters, and you've also got the data about how those batches go, very quickly, and this has started to happen already, when you apply AI to that sort of bioprocess, it can very quickly draw out non-intuitive insights, where again, we might miss it just because there's like 20 different parameters and we don't know where certain bumps or increases in yield or quality might be coming from. But when you combine together a host of really good quality data that is connected with the end results of a batch process, and you use these kind of AI machine learning algorithms and programs on it, I anticipate that if more and more people were doing it, they would find some pretty substantial reductions in COGS. And I don't see it happening as much as people should, I don't think. It's a bit expensive to implement it on the get-go, to look at these additional process analytes and whatnot. But in the long run, I'm convinced that that sort of investment will yield some substantial savings in the long run.
Oh, yeah, absolutely. And I think we're just at the very beginning. There's so much potential with all these technologies and learning we're going to make along the way. Now, let's look ahead, because I always love to ask this question about the future. Obviously, we don't know a lot of things about AI, but still, I'd love to have your take on that. What do you believe will be the most significant changes in cell and gene therapy over the next decade?
Good question. Inside of gene therapies, I got my start doing gene therapy and made AAV in the lab, so I'm a little bit biased. I love it. It's a nifty little vector. It's done a lot of good, but there are some pretty substantial advances that are coming from the non-viral side with LNPs and whatnot. And so I anticipate them continuing to kind of eat away at AAV's market share, although I don't think AAV is going to be going anywhere soon. So I do see that coming in. LNP is just kind of starting to take a bit more of a larger chunk of the market. With regards to cell therapies, there's two different paths, right? There's the autologous ones where you're taking cells from a patient, transforming the cells and then putting them back into the same patient, versus allogeneic where you're taking a bank of cells and you're just kind of divvying those out to everybody, where it's kind of a bit more of a universal treatment. Allogeneic is definitely going to increase its market share. However, I think in terms of cost reduction, one of the big players inside of the space that people aren't quite appreciating the way that they should, although they've gotten a lot of attention, is someone like Cellaris, where they're able to basically automate the entire process and really reduce that variability in between batches. And so I think it still remains to be seen as to how effective that'll be across just a wide combination of different autologous therapies. But in terms of allogeneic, like it's a match made in heaven. If allogeneic cell therapies can get rid of the problem associated with like graft donor rejection and that sort of thing, then I just see that as a chart that's really going to be very effective in terms of reducing the cost per dose. If you kind of remove the logistics of the whole thing, you've got a centralized cell bank. Someone has this cancer in Kansas and the treatment center is in, I don't know, Chicago or something. Very quickly, that batch can be made for that person using an automated process. The amount of FTE is quite low. Therefore, the cost to the patient is quite low. I think that's probably one of the fastest routes towards reductions in costs for allogeneic. And so I think that the market will react accordingly. That'll be one of the big changes we see in the next 10 years. I predict to some extent that cell therapies will see a greater reduction in cost than gene therapies, at least for right now, but we'll see.
And beside the cost per dose, what other challenges does the industry face and how could we overcome or how could we address these challenges?
Aside from costs, I would say gene therapies, again, are a bit more of my skill set, even though I've spent some time in the cell therapy space. With regards to gene therapies, one of the big issues right now is quality, specifically with AAV, where when the field first got its start, it was like, okay, all we need to look for is the number of vector genomes, and we really don't care about empty capsids that are getting into the prep. The concerns about safety just weren't as well appreciated until several kids died because a large amount of empty vector along with the therapeutic dose that was needed, which was quite high, just kind of overloaded their livers. That was a tough blow for the industry. It's like, oh, shoot, we can't just leave these empty vectors and think they're not going to do anything. They are a contaminant that can cause all sorts of strain. And so people started to characterize the full empty ratio and started to optimize towards that. But now what we're starting to see even more is full and empty isn't good enough. Because what we're seeing now is not only are there full vectors that have your full therapeutic transgene inside of them, and you've got your empties that have nothing inside, but there's intermediates, there's partially full vectors that can have all sorts of stuff in it. It can be a truncated portion of your transgene. It can be host cell DNA. It can be backbone DNA. It can be a bunch of different things that aren't characterized and you're sticking them inside of a patient. And so my understanding from the antibody space is around 20 years ago, the FDA really cracked down hard being like, hey, you've got this antibody treatment, but you've got to characterize any DNA or any sorts of genetic material that's inside of your final prep. So how much worse is it now that you've got a protein case that's protecting uncharacterized DNA that you're now specifically designing to go inside of a patient's cells and then crack open? So I anticipate that in the next, I don't know how long it's going to be, but I think the FDA is going to come down pretty hard on this. You're going to have to characterize what's inside of your capsids, and you can't really be putting large amounts of anything that's not your transgene inside of people, because again, putting uncharacterized DNA inside of people is not going to be a viable strategy. That is not good. So we'll see what happens, but I do anticipate the FDA coming down on that sooner rather than later.
Oh, yeah, absolutely. I mean, it's as with all kinds of drugs, it's a risk-benefit ratio. And I mean, this has to be all right. I mean, we cannot afford having people dying just because we have a life-saving therapy. I think there's definitely room for improvement. Well, the cell and gene therapy space is evolving and a lot of exciting advancements are going to happen. What advice can you give young scientists who want to work in that exciting field, and also, like, in particular, how to stay updated on all the amazing advancements?
It's a good question. This is going to sound a little bit biased given the earlier part of our conversation, but more and more, LinkedIn is where it's happening, man. That's where you can connect with people who are already inside of the industry. That's where you can stay best up to date with regards to everything that's going on. The last several jobs that I've gotten have come straight through LinkedIn. And so in terms of staying up to date and breaking in, LinkedIn is the best place to do both of those. And again, I'm a little bit biased here, but It's such a powerful method of creating genuine connection with people that you would have never met geographically otherwise. Unless you're living directly in Boston and you just happen to bump into people on the street, or you're going to the same restaurant. That's basically what can happen on LinkedIn. So if you're interested in breaking into the cell and gene therapy field and you're not quite sure how to do it, LinkedIn is a great place to get started, to start engaging with different folks in this area. To not just ask people for stuff without anything in return, but to provide some value up front, to do some work like reading a paper for them ahead of time and then posting your insights. It's a way to connect with people you'd never meet otherwise and ask for a 10-minute coffee chat, be it virtual or in person. And so, yeah, LinkedIn is a great place to get started and to meet some people inside of the field.
Yeah, I totally agree. Just to give you one specific example of smart biotech scientists, that's how Ben and I met. It's over LinkedIn because we started to engage. I think I noticed your content. I put some comments on your posts, and everything started from there. We sent a few messages, and now we're having a podcast interview. And we have never met. So that's the amazing opportunity you have on online platforms to meet amazing scientists from all over the world and connect.
It's so cool, right? Like, yeah, we've never met. At one point, you lived around 20 minutes away from where I live. But aside from that, we're in different countries across the world, on different sides of the world. And yet through LinkedIn, this is the only way that this has ever happened.
Exactly. And to circle back to one of the points we mentioned at the beginning of our conversation, it's important to connect with people. And I think that's what makes it so powerful. Now, we have covered a lot of ground today, Ben. What do you think is the most important takeaway you want our listeners to walk away with?
I would, again, recommend that if you're not using LinkedIn, use it a little bit more, just in terms of being able to stay up to date on the industry. On top of that, I think that if there's people who are listening to this who work in bioprocess, reconsider how you're using process analytical technology, especially with the advent of artificial intelligence. There could be huge benefits that you could gain from just starting to get a really solid, well-curated data set about your bioprocess. And a great way to start with that is use process analytical technology. Find out what are some of the key parameters that you're not looking at now that you should be, and start looking around to see how you could potentially use AI to analyze your process and ultimately save your company a bunch of money, because there's definitely savings and yield appreciations that can be found by combining those two things together. And most people aren't doing it yet. So if you're a CDMO or a CRO, that could be a huge competitive edge.
Fantastic. Wonderful. This is a great way to summarize our conversation. Ben, where can people connect with you? And I'm already expecting the magic word, but I'll let you tell it.
Well, thank you for asking, David. I can be found on LinkedIn, available at app stores near you. That's the major social media I've used. I haven't really branched out into anything else so far. I might eventually, but that's definitely the best way to contact me.
Awesome. In case you have not heard it yet, it's LinkedIn, by the way. Well, thank you so much, Ben, for sharing all these insights. It was a huge pleasure having you on the show today. Thank you so much.
Glad to be here. Thanks so much, David.
All right, smart scientists. That's all for today on the Smart Biotech Scientist podcast. Thank you for tuning in and joining us on your journey to bioprocess mastery. If you enjoyed this episode, please leave a review on Apple Podcasts or your favorite podcast platform. By doing so, we can empower more scientists like you. For additional bioprocessing tips, visit us at smartbiotechscientist.com. Stay tuned for more inspiring biotech insights in our next episode. Until then, let's continue to smarten up biotech.